So I want you to imagine a patient. She's sitting in her oncologist's office, and she's completely exhausted. She has metastatic breast cancer, and her latest blood work shows severe anemia.
Right, which is incredibly common, unfortunately.
Exactly. Her circulating red blood cells are basically starving for iron. So naturally you'd think, okay, she just needs an iron infusion, right? Like just give her more iron.
Right, that's the logical leap.
But then if you take a closer look at her bone marrow biopsy, there's this massive paradox. Her body is actually hoarding vast amounts of iron right there in the marrow.
It's just sitting there.
Yeah, it's stockpiled in these really heavy concentrations. But her red blood cells, they can't access a single drop of it. It's as if the entire supply chain has just been hijacked.
And, that paradox, the body hoarding this vital resource while the blood literally starves, is exactly what this groundbreaking 2025 paper in the journal Cell finally explained.
Oh, wow. Okay.
Yeah, the researchers uncovered that the tumor isn't just passively taking up space in the bone. It's actively manipulating the local immune system to reroute the body's iron supply directly into itself.
Okay, let's unpack this. Because today we are doing a deep dive into this incredibly fascinating study. It's titled Tumors Hijack Macrophages for Iron Supply to Promote Bone Metastasis and Anemia.
It's a heavy title, but a brilliant paper.
It really is. And our mission today is to completely decode how tumors pull off, well, what I can only call the ultimate biological heist.
That's a great way to put it.
Right, because they steal the body's iron supply to build their own survival armor against this harsh environment. And they do all this while systematically starving your red blood cell factories. By the end of this deep dive, you're going to look at cancer not just as this rogue mass of cells, but honestly as an incredibly cunning master of disguise.
Yeah, we really have to fundamentally shift our perspective here. Metastatic cancer is not just a rogue multiplier. It functions much more like an invasive architect, I'd say.
An invasive architect. I like that.
Right. So when breast cancer cells migrate to the bone marrow, they encounter an environment that is deeply hostile to them. And that is primarily because the marrow is highly hypoxic.
Hypoxic meaning really low in oxygen.
Exactly. The stem cells that naturally live in the marrow, they evolve to thrive in that low oxygen tension. It actually helps preserve their stemness, their ability to become different types of cells.
Okay, makes sense for that.
But a breast cancer cell, that cell is accustomed to the highly oxygen-rich environment of mammary tissue. Technically, it should just suffocate and die in the bone marrow.
So keep that hypoxia detail front and center in your minds, everyone, because that right there is the ultimate motive for the heist. The tumor is suffocating.
Precisely.
But before we look at the crime itself, let's map out the normal factory floor of the bone marrow. Because we need to know what normal looks like, right?
Yeah, you have to know the neighborhood.
Exactly. So deep inside the marrow, we have these specific functional zones. They're called erythroblastic islands, or EBIs.
Right.
And these are essentially the maternity wards where mature red blood cells are born. They come from these baby precursor cells called erythroblasts.
And those erythroblasts, to grow up and do their jobs, they require a massive continuous influx of iron to build hemoglobin.
Because without hemoglobin, a red blood cell is useless.
Yeah.
Right? They can't carry oxygen.
Exactly. It's just an empty shell. But here's the catch. Erythroblasts cannot efficiently scavenge free-floating iron on their own. They just aren't good at it.
Oh, really?
Yeah.
I always assumed they just sort of soaked it up.
No, they rely entirely on a central nurse cell to do it for them, specifically the EB macrophage.
Okay, the EB macrophage.
Yeah, and the researchers isolated these crucial macrophages by identifying a specific triplet of protein receptors on their surface. It's a VCAM1, CD163, and CCR3.
Okay, wait, let's actually visualize what those protein tags do rather than just throwing like an alphabet soup of acronyms at the listener.
Fair enough, yeah.
So let's picture the erythroblastic island as a highly regulated hospital cafe. It's
Yeah.
super busy. The EB macrophage, the nurse cell, is your incredibly dedicated, highly specialized barista.
I love this analogy.
Right. So the baby red blood cells, the erythroblasts, are the exhausted hospital workers crowded around the counter. And the iron molecules are the essential espresso shots they desperately need.
Absolutely.
Now, those tags, that CD163 tag on the macrophage, that is the scavenger receptor. That's how the barista actually pulls in old coffee beans and, or in this biological case, recycles iron from dying red blood cells.
Right. It's the cleanup and prep crew.
Yeah. And then the VCM1 tag, that is the physical counter itself. It's an adhesion molecule that literally lets the macrophage hold hands with the baby red blood cells to make sure the iron is transferred safely and directly.
And the precision of that transfer is vital, right? Because free iron is actually highly toxic to tissues.
Wait, iron is toxic.
Oh, yeah.
Okay.
If it's just floating around freely, it generates reactive oxygen species, which basically fry your cells.
Oh, wow. I had no idea.
Yeah, so the EB macrophage safely stores that recycled iron internally, and then it uses a very specific exporter channel to hand it over safely directly into the waiting erythroblast. It's this beautiful, closed, perfect loop.
Okay, so let's do a quick bulleted recap of our baseline here, just so we have the foundation locked in before the invasion starts.
Let's do it.
Bullet 1, we have a hypoxic, low-oxygen marrow factory. Bullet 2, we have baby red blood cells, the erythroblasts desperate for iron. And bullet 3, we have these nurse cell baristas anchored by VCAM1, safely recycling and handing over that iron via CD163.
Perfect summary.
So now I want you, the listener, to picture that highly organized, peaceful cafe. What happens when metastatic breast cancer just crashes into this delicate ecosystem?
Well, the architecture is completely dismantled. It's chaos.
How did the researchers actually see this happen?
They utilize these specific murine mouse cell lines called E0771 and Pi811 Elaine.
Okay, mouse models. Why those specific ones?
Because they are immunologically intact and they aggressively mimic how human breast cancer hunts for bone tissue, they're a really good proxy for the human disease.
Got it.
So when they introduced these tumor cells and observed the marrow, they found something crazy. The tumor physically recruits those iron-rich nurse macrophages away from the baby red blood cells.
It literally drags them away.
Yes, it pulls them out to what we call the tumor stroma interface, which is basically the outer perimeter of the cancer mass.
Wait, I need to push back on the mechanics here for a second.
Sure.
If the tumor is aggressively pulling in all these macrophages and nutrients, and it's rapidly dividing at this outer interface, wouldn't standard chemotherapy, which specifically targets rapidly dividing cells, wouldn't it just nuke the tumor right there? Why does this specific iron theft make it so untouchable in the clinic?
And that right there is the crux of the entire problem.
Okay.
Standard chemotherapy targets the mechanics of active cell division, like DNA replication. But remember that hypoxic, low oxygen environment of the bone marrow.
Right, the suffocating environment.
Exactly. That hypoxia naturally forces a lot of cells, including these invading tumor cells, into a much slower dividing, almost dormant state initially.
Oh, I see. So they aren't dividing fast enough for the chemo to notice them.
Exactly. The immediate threat to the tumor isn't dividing, it's suffocating. It needs to survive first. And to survive, the tumor executes this highly specific molecular handshake to steal the iron.
Okay, tell me about this handshake.
So remember the macrophage? It has an iron delivery chute called FPN, or ferroportin. It's actually the only known iron exporter in vertebrates.
So the barista is standing there, holding the espresso, with the FPN delivery chute wide open, totally ready to serve the baby red blood cells.
Right. But the tumor cells dramatically upregulate a different receptor on their own outer membranes called CD71.
CD71, what does that do?
It's an iron importer. So the tumor essentially builds a counterfeit docking station that fits perfectly with the macrophage's FPN chute.
Oh, wow. It's literally hacking the delivery system.
Yes. The macrophage opens the chute, thinking, hey, I'm feeding an erythroblast. But the tumor is using that CD71 vacuum to pull all that stored iron directly into its own cytoplasm.
That is wild. And the researchers, they actually proved this physical transfer visually, right? Like they didn't just infer it from some spreadsheet of genetic sequencing.
Yeah, they captured it visually, which is so cool. They used something called Prussian blue staining.
Prussian blue. Like the paint color.
Yeah, basically, it's a classic pathology stain that binds specifically to stored iron, and it turns it this brilliant, unmistakable blue color.
Okay.
So under the microscope, the researchers saw these bright blue, incredibly heavy macrophages cluster intimately right against the tumor cells.
Like the barista's being held hostage against the tumor wall.
Exactly. And over a measured time course, they watched as the brilliant blue stain inside the macrophages slowly faded, while the tumor cells themselves began to stain deeply blue.
Oh, wow.
Yeah, they caught the iron moving across the interface in real time.
Okay, let's lock in this mechanism with our mini recap because this is dense, but it's so important. Bullet 1, the tumor invades the marrow. Bullet 2, it drags the nurse macrophages away from the red blood cells. And bullet 3, It builds a counterfeit CD71 receptor to hack the FPN delivery chute and suck the macrophage dry of iron.
Spot on.
And, this brilliantly explained the clinical paradox we opened the show with. The anemia isn't just some random byproduct of being sick, right?
Not at all.
The erythropoiesis, the creation of new red blood cells, it halts completely because the iron supply chain has been physically rerouted to the tumor.
Exactly. And this is why those patients exhibit high serum ferritin, which tells us the body is stockpiling iron at the scene of the crime, but they have really low circulating serum iron.
Because none of it is actually reaching the blood. The erythroblasts are just starved out of existence.
Yep. They just die off.
Okay, here's where it gets really interesting. Because, sure, we know the tumor is stealing the iron, but iron isn't just a generic nutrient like glucose. Why go to such incredibly elaborate lengths to hoard iron specifically?
Because the iron is the literal key to surviving that hypoxia. The tumor takes that iron and engages in a process the researchers call erythroblast mimicry.
Erythroblast mimicry. Wait, so the breast cancer cell is literally putting on a disguise and mimicking a baby red blood cell?
Yes.
But how? How does a breast cell suddenly, remember how to act like blood? It's from the breast.
It does it through epigenetic reprogramming.
Epigenetic reprogramming. Let's break that down.
So every single cell in your body contains your entire DNA blueprint, right? But in a breast cell, the specific genes for blood functions are tightly schooled up and locked away. They're silenced.
Because a breast cell doesn't need to act like blood.
Right. But when the tumor pulls in this massive influx of stolen iron, that iron acts as a signaling cascade. It alters the chemistry inside the cell. And that iron influx specifically allows the tumor to upregulate a transcription factor called GATA1.
GATA1.
Yes, and GATA1 is the master genetic switch that red blood cells normally use to produce hemoglobin.
Oh, goodness. So the iron literally alters the internal chemistry of the breast cancer cell, unspools that dormant, locked away DNA, and allows it to read the recipe for hemoglobin.
Exactly. And what's fascinating here is the sheer evolutionary plasticity of the cell. By turning on the hemoglobin factory inside itself, the breast cancer cell can bind whatever tiny trace amounts of oxygen exist in that hypoxic bone marrow.
It's building a scuba tank.
Yes. It synthesizes its own internal scuba tank. It steals the iron from the blood to build the exact protein the blood was supposed to build just to protect itself from hypoxia-induced cell death.
Okay, quick synthesis of this mimicry bullet point style. The tumor steals iron. The iron unlocks dormant DNA via the GATA1 switch, and then the breast cell builds hemoglobin so it can breathe in a suffocating environment.
That's the mechanism.
It's quite literally identity theft at the genetic level.
It really is.
But, okay, I have to play the skeptic here for a minute. Because I buy the lock and key mechanics of FPN and CD71, and I buy the epigenetic unspooling. But mouse genetics are highly controlled. They're manipulated in a pristine lab. Are we really seeing human breast tumors act like biological vampires, pulling off this identity theft in real patients?
That is a great question, and the clinical validation in this paper is arguably its absolute strongest component.
Okay, lay it on me.
The researchers took tissue biopsies from actual human bone metastases across multiple different cancer types, not just breast cancer, but lung and kidney cancers too.
Really? Lung and kidney as well?
Yeah. And when they applied those iron stains to these human samples, they found the exact same architecture. Dense clusters of iron-rich EB macrophages physically tethered to the tumor cells actively feeding them.
So this barista hijacking, it's not just a breast cancer thing. It's a universal survival strategy across different human cancers.
It certainly appears to be, yes. But honestly, the most striking human data came from analyzing primary breast tumors.
Primary, meaning the original tumor mass in the breast, right?
Yeah.
Long before it has spread anywhere.
Exactly. Looking at a huge cohort of over 600 human patients, they discovered something incredible. If a primary breast tumor shows a high expression of that hemoglobin gene, the HBB gene, it serves as a massive independent predictor that the cancer will specifically metastasize to the bone later on.
Wait, hold on. The tumor is already turning on the hemoglobin genes while it's still sitting in the highly oxygen-rich breast tissue.
Yes.
It's like it's practicing holding its breath before it even makes the journey to the bone.
That's a great way to think of it. The cells within that primary tumor are highly heterogeneous, right? They are constantly mutating. And the specific cells that randomly happen to unspool that GATA1 pathway and figure out how to make hemoglobin, those are the exact cells that are perfectly pre-equipped to survive the journey through the bloodstream and colonize the hypoxic bone marrow.
Wow. They are naturally selected for metastasis.
Exactly. It's pure Darwinian selection happening inside the tumor.
That is terrifyingly elegant. Okay, let's
Yeah.
do our final mini recap before we talk treatments. We know the mechanism, we know it happens in humans, and we know it actually predicts metastasis. So how do we stop the heist? Did the researchers figure out a way to break this supply chain?
They did, and they used some incredibly sophisticated bioengineering to prove it, to show that these specific nurse macrophages were the absolute linchpin of the tumor's survival. They used an engineered mouse model called the JDI mouse.
JD. Like Star Wars. Just EGFP death-inducing T cells. It sounds like science fiction. How does that system actually work?
It's brilliant. Normally, T cells hunt their targets based on complex antigen presentations, which tumors can easily evade or hide from. But in the GDI system, the researchers genetically engineered those specific iron-recycling macrophages to express a green fluorescent protein, or EGFP.
So the macrophages literally glow green.
Right. And then the mice are equipped with these specialized killer T cells that are hardwired to act as biological hitmen. They bypass all normal rules and just seek out and destroy absolutely anything that glows green.
Okay, so they gave the T cells laser focus to assassinate only the corrupted baristas.
With absolute surgical precision. And when the GDI T cells depleted those specific EB macrophages, the results were astounding.
What happened?
Without that iron supply line, tumor growth in the bone just plummeted. The cancer cells completely suffocated.
Yeah.
And, just as importantly, the systemic anemia improved.
Oh, really?
Yeah, because the tumor could no longer hoard all the iron, so the normal baby erythroblasts could finally get their espresso and resume their normal jobs making blood.
Wow. But obviously, we can't deploy fluorescent hitmen T cells in human patients right now. Did they validate this with, accessible therapeutics, drugs we actually have?
They did. They utilized diferoxamine, which is a clinical iron chelator. It basically acts like a chemical sponge that just mops up free iron in the body, completely depriving the tumor of the raw material.
Okay, so a sponge.
Yeah.
Yep. And they also genetically knocked out the FPN iron exporter on the macrophages, which is essentially like welding the delivery chute shut. And in both of these interventional models, tumor progression was halted.
Man, if we connect this to the bigger picture, this fundamentally changes the clinical paradigm for treating cancer-associated anemia, doesn't it?
It necessitates a massive reevaluation, yes. Think about it. A metastatic breast cancer patient presents with severe anemia. The traditional, totally reflexive clinical response is to administer an intravenous iron infusion or prescribe heavy iron supplements to try and boost their red blood cell production.
But based on everything we just decoded, if you pump that patient full of IV iron without first blocking the CD71 receptors on the tumor, where is that iron actually going?
It is being completely intercepted by the tumor.
Oh my God.
Right. You are not treating the anemia at all. You are literally handing the tumor the exact building blocks it desperately needs to forge more hemoglobin armor and accelerate its own growth. You are actively feeding the metastasis. Wow. Okay, so what does this all mean? Let's pull all the threads together for a final synthesis for everyone listening. We started with this huge medical paradox. Patients whose bodies are hoarding iron while their blood starves.
Right.
And we discovered that metastatic tumors don't just brute force their way into the bone. They act as these invasive architects. They physically kidnap the EB nurse macrophages. They hack the FPN delivery chute using their counterfeit CD71 receptors.
Exactly.
And they hoard the iron to trigger this wild epigenetic unspooling, allowing a breast cell to mimic a red blood cell and build a hemoglobin scuba tank to survive the hypoxia. And finally, the most chilling part. If we don't recognize this disguise, our standard treatments, like giving iron, might actually be fueling the fire.
It really is a masterclass in tumor plasticity. The cancer learns the exact metabolic rules of the tissue it is invading, and then it perfectly exploits them. The microenvironment isn't just a backdrop, it is a highly active participant in the disease.
That is just incredible. And that leads me to a final provocative thought for everyone listening today. We just spent the last 20 minutes exploring how a breast cancer cell can completely adapt to the bone marrow by literally mimicking a red blood cell. It alters its own epigenetic identity to survive.
Yeah.
If cancer is this adept at shape-shifting, what on earth is it mimicking when it metastasizes to the liver or when it breaches the blood-brain barrier? I mean, I know there are emerging studies looking at things like vasculogenic mimicry in the brain, where tumors literally pretend to be blood vessels.
It's a huge field of study right now, and it forces us to ask a really uncomfortable question. Are our traditional chemotherapies failing because we are still stubbornly treating the original breast cancer cell, completely ignoring the highly specialized disguise it has adopted in its new home?
Because if a breast tumor is dressed head to toe in the molecular clothing of a brain cell, or a liver
Mhm.
cell, or a red blood cell, our targeted drugs might just walk right past it.
Exactly. They won't even recognize it as the enemy.
So I want to pose a question directly to you, the listener. What stands out to you about this survival mechanism? Does thinking about cancer not as a greedy lump, but as a shape-shifting structural engineer change how you view disease? I really encourage you to mull over just how clever and terrifying cellular adaptation can be. How much of medicine is simply learning to see through the disguise. Until next time, keep questioning what you think you know.